Thank you, everybody, and good evening it's really nice to have all of you guys to join us on a Saturday night at 8 o'clock, and then for scientific webinar. I know it's like everybody's so devoted and dedicated to our webinar, and also thank you both the speakers. So I want to give a really quick introduction about our organization, which is International IVF Alliance, funded by a group of embryologists and also REI physicians. So I am your host. My name is Yifei Liu. I work at Penn State. I'm the IVF lab director. And also want to thank you three our members, Wei Zhang, Liang Yu, and Yilin Li. They've been helping me organize the webinars, being tremendously helpful. So I'm really appreciative and grateful. So I also want to give an introduction of our two speakers tonight. I used to be one, but tonight we have two. So this is a bonus, I would say. Dr. Jesse Major is a professor in the Department of Vet and Animal Science at UMass Amherst and serves as the Associate Vice Chancellor for research support. His lab has long-standing NIH funding to characterize early lethal phenotypes generated by the KOMP2 as a knockout mouse project. I don't know if there is a the new version of KOMP. I used to work on the KOMP1. So it's kind of exciting.
Uh-oh, I think we lost Yifei again.
Yifei, your another computer is working.
Hello, can you hear me?
Yeah.
Yep, now you're back. Nope, he's gone.
It has two computers, I think.
Perhaps I will introduce Doris and we proceed, or should we wait for Yifei to jump back in? Okay, well, hello everyone, I'm Jesse Major. I'm beardless today, but bearded in the picture, but that's still me. Our second speaker is Doris He, who just graduated from my lab with her PhD. And so she's a newly minted doctor. As you'll see, she's done some excellent work on early pre-implantation development, which has led to some really exciting discoveries, which she's going to tell you about. Her background is she has worked as an embryologist at NYU IVF for a few years before she came to my lab to study really early gene function. And she's really interested in, as you'll see, in bridging clinical and sort of basic research science toward improving IVF and ART outcomes.
All right, thank you so much, Jesse.
Sure. Oh, you're back. Excellent.
I'm back. So, thanks for helping me out. I don't know why tonight is just like, does not work well. So I will just, let you guys take it over from me, okay?
Okay, great.
Thank you.
So I will share my screen and we'll get started. Hopefully you're all seeing my screen. Let me put on a laser pointer. Can you see my mouse as well? I hope, laser pointer? Yeah.
Yes.
Great. Okay, so as I said, I'm Jesse Major. I'm a professor in the Animal Science Department at UMass Amherst. This is a picture of our current group in front of our beautiful, lovely new building. And my whole research career, I've studied these things over here to understand this thing, right? So this is really the first third of mouse embryo development that I'm showing you here. Both pre-implantation and gastrulation stages is really what I've always focused on. And really, this is a model for human development, as I think you can all appreciate. So I think most of you on this call know better than me what human embryos look like. I've always worked in the mouse, although recently with Doris's project, I've learned how to handle human embryos as well. But if I gave this to you and said, okay, which one's human, which one's mouse? Who can recognize which is which? I'd be pretty hard pressed. I think some of you experts might be able to tell, but the point is they're really identical, right? These early stages of development are nearly identical, both in morphology and in the gene function at these very early stages. So we have human on the top here, mouse on the bottom. Not only is pre-implantation nearly identical morphologically, but the next few stages are extremely similar as well. So that's right around implantation, although the timing between mouse and human is different. The processes are very much the same. Both pre-implantation, implantation, and the very next couple days following implantation, the same morphogenetic movements happen prior to gastrulation. Human and mouse gastrulation diverge somewhat morphologically. We know relatively little about human gastrulation because it's a very difficult time to study for various reasons. Just getting samples and doing experiments is nearly impossible, which is why the mouse has really become the preeminent model for understanding early development, both genetics and working out sort of new twists and turns on reproduction and successful development. As you know, I think our genetics are very similar with the mouse. We share nearly all the same genes, just with some minor variations. And that's why about 15 years ago now, actually probably more like 18 years, the Knockout Mouse Project was initiated. And this grew into what's now termed the International Mouse Phenotyping Consortium. And this is really a global effort. You can see on the world map here a bunch of different centers, these little blue circles. It's a combined effort to make a null mutation or to knock out every protein coding gene in the mouse. So let's just let that sink in for a minute, right? There's about 20,000 genes probably. And the goal here is to make a mutation in each one separately, really to understand gene function, right? And so this is, they call this the first comprehensive functional catalog of the mammalian genome, right? We know the human genes, we know the mouse genes, they're very high degree of overlap, and we're asking what's the function of every gene in the mouse. The main goal of the IMPC is really to look at adult phenotypes. So they make knockouts, and they have a huge battery of tests that they put all of these knockout animals through, largely looking at biomedically related function. And in this way, they're able to identify, oh, these 200 genes are involved in eye function. These 400 genes are involved in cardiac function. These 600 genes all have something to do with neurobehavior. So it's a very powerful tool to look globally across the genome and see what genes are involved in what functions that may be very relevant for human disorders and diseases that we deal with. What was known going into the design of this project is that a pretty high percentage of genes are essential, right? So this is a little outdated, but the numbers, this was from a year ago, when they had assessed about 8,159 different knockout genes. What they found was that 24% of these knockouts are lethal, meaning you cannot make a mouse that's missing any of those genes. So it's a pretty big number, right? It turns out that half of those that are lethal at weaning are what's called early lethal, meaning these genes, when you knock them out, they cannot even find embryos at day 9.5 that are homozygous for that mutation. 9.5 is the stage just after what I've shown you here, right? So these half of the lethals, which is 12% of all genes, are required for pre-implantation or gastrulation stages. And over the last 10 or 12 years, my lab has really become the only place that we're characterizing these early lethal alleles. So we've been fortunately funded to look at a large cohort of these knockout lines made by the IMPC that all have an early phenotype. And so we've had to develop a sort of phenotyping pipeline that we use in our lab, because as you'll see, to date, we've characterized more than 250 different genetic knockout lines, which means we have to import mice for each one, make a small colony, set up timed heterozygote by heterozygote matings so that we can do carefully staged analysis of embryos at different times. And so our pipeline looks something like this, where we'll start with heterozygous intercrosses. We first dissect at day 7.5, which is right in the middle of gastrulation. And we can have a few different outcomes here. So sometimes we find the embryos and they look normal. So we'll look a day later. Sometimes we find the embryos and they look abnormal. So we'll do a bunch of analyses on day 7.5 and day 6.5. And sometimes we fail to find the embryos at day 7, and that's when we back up prior to implantation and dissect at day 3.5, where we expect to find blastocysts, and we do the analysis at that point. So this is our overall workflow, and we've got it down pretty efficiently so that each mouse line we get in, it takes between 6 and 9 months from breeding to mating to doing all this work to figuring out where the mutant embryos are and what they look like. And we perform some basic analysis on every single mutant that we largely make public, as you'll see in a minute. So as I said, we're going through quite a large number of lines. Each of these little pictures behind is one different knockout line and one litter of dissection from that knockout. So tons and tons of embryos. My joke in my lab is that our hashtag is all embryos all the time, because it's kind of all we deal with, lots and lots of embryos, which makes for a lot of fun. So here's one example. This is a gene called MED20, or mediator complex subunit 20. And I'll be honest, when we first got into this work, I tried to be very inclusive, meaning I tried not to look at the gene and say, I think that's interesting, I think that's not interesting. Because the idea is to catalog all gene function. And it led us to things like this. So MED20, if you remember from your probably either high school or college textbooks, mediator complex is supposed to be in every cell. It's a very basal function, and I would have predicted it to be just cell lethal and not very interesting in terms of linear specification or really any exciting findings. So here's a litter from a het-by-het cross, both heterozygous from MED20. I'm showing you 3.5 because this is a line where we could not find any mutants at day 7, so we backed up to flush blastocysts. And you're all embryologists here in the audience. If I could see your hands, I'd say, okay, someone tell me which are the mutants. I've masked their identities with these black bars, right? Probably most of you are going to look at these embryos and say, well, there's something going on with these 3 over here. Turns out those are not at all the mutants. Those just happen to be a wild type and a few hets that are a little delayed. And so for this reason, in the mouse, certainly we have these big litters. We have to genotype every single embryo that we can in order to feel confident of which are the mutants. This is all work that a previous postdoc of mine, Wei Kui, performed, did a great job characterizing, and we can see the mutants for MED20. Here's one here, here's one here, here's one here. They look quite nice, right? Perfectly normal morphologically. But I'll remind you, we cannot find them at 7.5. So something's going wrong during implantation. Since implantation is extremely difficult to capture in vivo, very laborious, we first start with it's sort of an in vitro surrogate assay for implantation. We call this an outgrowth assay. And if you take a wild type embryo, so maybe this one, and you put it in the right conditions in our incubator, what happens is that blastocysts will attach to the plate and you'll get this nice inner cell mass, or now an embryonic stem cell colony outgrowth in the middle, surrounded by the trophectoderm, which you can kind of see all around the plate with these giant nuclei, if you look closely. So I'm showing you here a wild type, a heterozygote, and a mutant. What's hopefully pretty obvious is that this MED20 mutant completely fails in this process. In fact, it doesn't even hatch from the zona pellucida. So it's a complete failure. You can still see that zona on this embryo right here. So Wei didn't stop there. He did what we often do, which is then start to look at lineage allocation and proper transcription factor assignment to different compartments. And so what I'm showing you on the top here is the first lineage decision, where we can mark 2 genes, OCT4 and CDX2, which are expressed in the inner cell mass and the trophectoderm, respectively. He's also looking for cell death with P53. And what you can appreciate is that the mutant down below looks perfectly normal. You get this beautiful mutually exclusive expression between OCT4 and CDX2. Nothing going on, no cell death, looks pretty good. He then looked at the second lineage decision, and this is where things start to get interesting. So we're doing different markers now. SOC17 marking the primitive endoderm, NANOG marking the epiblast, again CDX2 marking the trophectoderm. So again, showing you wild type, heterozygote, and mutant embryos. And hopefully what you can see is that although SOC17 looks fine, the NANOG expression in the mutant is misexpressed in this trophectoderm lineage, telling us that these outer cells are really pretty confused. Wei backed this up by doing some RNA-seq analysis and some other types of assays, which really told us that this TE lineage was not being specified correctly. And that actually fits really nicely with the fact that they fail to hatch from the zona, because it's these outer cells, the trophectoderm, which are required to perform that initial function of piercing through the zona, hatching, and then attaching to the uterine lining. So that nicely confirms or sort of makes a nice story about why Med20 is recovered at 3.5, looks pretty normal, but totally fails to implant, and we cannot find them at day 7. This is the type of story we like to generate. Wade took this a bit farther and published a nice paper, which you can read if you like. But for every single one of the different lines that we characterize, we also put the data back out to the public. So this is part of our funding work as well. We maintain our own blog where we display all of this data almost in real time as soon as we have gathered it. These pages are also linked through all of the mouse relevant databases like the IMPC and Mouse Genome Informatics, which you might be familiar with as well. So as you'll see in a minute, this is one main type of phenotype that we see, meaning an implantation failure. The other major type that we come across look like this. So this is a different gene. This is an MRPS25, or mitochondrial ribosomal protein. This is one that we can recover at day 7. And so here's a mutant embryo below. It's wild type or heterozygous littermate, which is starting to gastrulate. And for these types of lines, we perform lineage analysis and some cell death and cell division assays as well by doing serial immunofluorescence assays on embedded and sectioned embryos. And that's just what you're seeing here. So we've worked out protocols where we can do 2 or 3 rounds of 3 or 4 color immunofluorescence, followed by H&E, all in the very same section. And this way we can gather a whole lot of data from a relatively small number of embryos. In this case, hopefully you can appreciate here in this transverse section, while the control embryo has initiated gastrulation, which you can see with these green cells, the primitive streak down here, which are marked by T, the mutant embryo completely fails in this process and remains in this egg cylinder state. A lot of our early lines also came with a reporter gene LAC-Z knocked into the locus. So when we've had that, we also provide that expression data on our blog site, just as this baseline data to give back to the community. So this is a typical gastrulation failure. Again, these are the 2 main phenotypes that we see. And what I'm going to do in the next 10 or so minutes is I'm going to switch gears a little bit and not tell you about specific genes, but we're starting to think more broadly because we now have 250 different phenotypes. So what different questions can we start to ask with this big collection of early lethal mutations and information that we have? And so 3 different questions I want to run through pretty quickly. 1, can we predict phenotypes for gene families and complexes? In other words, do we have to do the analysis on every single early lethal? Or will we reach a point at which you tell me a gene, I can tell you what the phenotype is based on the information we already have? Second question, is there an even distribution of phenotypes across all these stages, which might be one prediction? Or are there specific developmental bottlenecks that we can identify even in these early 8 or so days? Thirdly, with this type of data we're generating, can we identify stage-specific pathways or networks amongst these new, using this new information that we're putting together? So to start, can we predict phenotypes? I'll take you through one example, which really started maybe 6 years ago. A student of mine named Agnes was starting to put together these big sort of tables for us, or visual tables of phenotypes. So each box is a different gene knockout. It's a little hard to see the names. And what struck Agnes was this set right here in the middle that all had a really identical looking phenotype. And not surprisingly, maybe they have very similar names. So here we have MRPL22, MRPL3, MRPL44, etc. And so she started looking into these further, and she published a couple of really nice papers characterizing these phenotypes, which I'll just very quickly summarize. So here are each of the mutants that she studied. Down below, a littermate control. Right? So MRPL3, 22, and so on. These are mitochondrial ribosomal proteins, just like the MRPS25 I just talked you through. And what was striking about these was that they reached this egg cylinder stage, which looked like a very normal, maybe day 5.5, day 6.0, right before gastrulation. Perfectly normal looking. And they just arrested there. Nothing further. And so these are all genes that are nuclear genes. And I'll remind you of some basic cell biology, that they're nuclear genes. They get transcribed from the nucleus, but then they're imported into the mitochondria, which is where they function. Right? And in this case, these are the mitochondrial ribosomal proteins. They fail to gastrulate. Agnes showed they have extremely abnormal mitochondrial morphology. So here's what a normal mitochondria looks like. You can see all the cristae, these little slivers. And in the mutants, she started calling these this salami phenotype. They have these big holes. And that had been seen before when mitochondria are really in bad shape and probably about to be resorbed by the cell. She showed that there was basically no detectable mitochondrial translation in these mutant embryos and extremely reduced ATP levels. But the mystery still remained of why they look so nice, right? You would think they would die. And what we figured out was that often when mitochondria have levels of dysfunction, that increases cytochrome C in the cell, which usually activates caspase 3, which leads to apoptosis. Pretty standard pathway that cells know to die. However, this process is ATP dependent. As I told you, these cells have almost no ATP. So we think they'd like to die, but they can't. So that's why they're left in this nice pristine state, at least at day 7. And we know by around day 9.5, the maternal environment does reabsorb these embryos and they're no longer present. So that's the overall phenotype. But this led Agnes to start thinking more broadly about this prediction possibility. And so here I'm showing you all of development, and she sort of gathered from all the literature all of the mitochondrial proteins that had been knocked out. And she started listing them underneath the stage when their phenotype occurs. We add the ones that we've characterized in my lab, and we can really see no correlation. I mean, there's this big spread, right? You have some that are pre-implantation lethal and some that are even viable, or very mild phenotype. So this was not very satisfying. What she started doing was thinking about it differently, and she started mapping each gene, keeping in mind the different phenotypes by color, onto a suborganelle level. So here's a cartoon of the mitochondria, right? You can remember this from your textbooks as well. And now we can start to see some correlations. So all the genes involved, for example, in the electron transport chain, have a gastrulation failure phenotype. All the genes that have been knocked out that are in the TOMs or in this the TOM and TIM pathway have a pre-implantation phenotype. So we think by drilling down to the suborganelle level, we can start to see and make some predictions. I feel fairly confident if you told me here's a gene that's involved in the protein degradation in the mitochondria, I would predict that's a gastrulation phenotype. So this is sort of a maybe result. We might be able to predict these mitochondrial functions. We have several other large groups and complexes like this. I'm not going to take you through all the details. I'll just tell you the sort of overall results. So for example, of these 250 phenotypes, we now have 10 polymerase II subunits. Invariant phenotype. Every single one of them looks like this. They make a morula, they fail to make a blastocyst. Maybe that's not so surprising, right? Polymerase II is pretty essential for a cell. So it's probably just a cell lethal phenotype, and it's probably maternal protein, which is getting them this far even. So that's a yes, we could predict in that case. Two other examples, we have a handful of genes in the Bork complex. Long story short, could not predict at all. Yet another complex of genes we've been studying are called the TAFs. Doris did some of this work. And there's two big protein complexes that work together, TAFs and SAGA. And it turns out that we can predict these phenotypes as well between pre-implantation and gastrulation. So overall, I would say we're not quite there yet, meaning we can predict some very clearly, but in general, we still need to look at the phenotypes. I'm actually okay with that because that'll keep us in business, right? It means we have to characterize lots more knockouts. So we'll keep proceeding. If we move to the second question, I've already alluded to the answer. So is there a distribution of phenotypes or are there specific developmental bottlenecks? A nice way to visualize this is in a simple pie chart. So here what I'm showing you are the percent of our knockouts that arrest at each of these stages as we move from basically fertilization, two-cell, morula, blastocyst, implantation, early egg cylinder, and then gastrulation. And I've just color-coded the slices of the pie representing the bigger percentages of the phenotypes that we see. So we have very few that are two-cell arrest. We have a handful more which make a morula but fail to make a blastocyst. Again, my prediction is that these are probably really cell-lethal phenotypes and that it's maternal protein and RNA that's getting us that far. We have a huge number that make a blastocyst, but then fail to be recovered at 7.5. And these come in two varieties. Some that fail in the outgrowth assay that I showed you, others that in vitro look pretty normal, but again, cannot be recovered at 7.5. So even though outgrowth might look normal in vitro, clearly something is missing functionally when you're in the uterus. Very few that I can dissect at 7.5, but do not make an egg cylinder. They're just sort of a little dying mass. But then this other really large category where so far we have about 76 genes that look very similar to the MRPLs that I showed you, where they make a nice egg cylinder stage embryo, but they fail to gastrulate. So take-home message here, there are very clear bottlenecks. And this honestly is what we predicted going into this whole project, meaning implantation and then gastrulation. Right? And we made that prediction largely because it was known that a fair number of knockouts can make a blastocyst, but then something goes awry when you try to implant. So that's when lineages really become important, that they're defined correctly. And it's not until implantation where the embryo is tested in this way. There's also other historic literature suggesting that you need to reach a certain cell number in order to begin gastrulating. And so we actually have some evidence that a very large number of these gastrulation failures really tie back into the cell cycle, and that they're actually just not dividing enough, or the cells are dying and they never reach that point in which they can start gastrulation, and that the maternal environment then reabsorbs them. So take-home message, clear, distinct bottlenecks around implantation and gastrulation initiation. I'll remind us here, given the context of this webinar, so this data is also telling us that 62% of early lethals are pre-implantation phenotypes. So across the whole genome, that's about 3,000 genes. Each of these is a potential marker for early lethality in humans as well. And we do know that there's pretty clear evidence that a large percentage of genes that are lethal in the mouse have similar phenotypes in human, although it's much harder to get that data, as you can imagine. Okay, so this third question of can we identify novel pathways with this new data? And so if we separate these genes, so I've listed here all the genes with early phenotypes on the right, all the genes with the gastrulation failures on the left. And if I use these genes as if they were an RNA-seq experiment or something like that, and I ask what pathways are represented among these gene functions, likewise separately among these gene functions, we actually do pull out very different results. It might be hard for you to read, but for example, the early phenotypes, the pathways and metabolic processes involved are much more to do with RNA processing and RNA stability. Right? And that kind of makes sense. We know early on in both mouse and human, and really all species, there's this huge switch that has to happen from maternal RNAs to zygotic RNAs, really taking over function. And so that's probably what that represents. What was a little surprising was that the gastrulation failures, the top hits here, have much more to do with translation and aerobic respiration. And so this is really letting us make some new hypotheses about what's going on here and why these processes are so much more critical during gastrulation than earlier. So long story short, we're starting to be able to identify stage-specific pathways and networks. We're looking forward to continuing this work and expanding our data set, which really will help us refine the answers to these questions. So with that, I'm going to quickly thank my whole big group and my funding, which largely comes from the NIH and NICHD specifically. And I will let Doris take it away and tell you about some more specific gene function and how and some very exciting recent work for really translating what we think into something meaningful for the clinical as well. So thanks for your attention. Doris, you're up.
Oh, thank you very much, Jesse. Yeah, let me try to share my screen correctly. You know, the first task I need to tackle tonight is try to share my screen correctly for the
You
audience.
can do it. We have faith.
Wait, let me see. I need to. There we go. Everybody can see my screen? Okay, without notes. Without notes, okay. All right, thank you very much, Jesse. And hi, everyone. My name is Doris. I just finished my PhD in the major lab. For those of you who don't know me, before I started PhD journey, I worked as a clinical embryologist. So as many of us know, that clinically, even in the best scenario, which according to the ASRM 2024 report, is a group of women who are under the age 35 with the use of PGTA, which is pre-implantation genetic testing for aneuploidy, has achieved the highest implantation rate, which is about 62.7%. Now, but this also means that even in this best scenario, nearly 40% of those euploid embryos still fail to implant. And the reasons behind this remains largely unknown. So in the field of IVF, implantation continues to be a major bottleneck, and this is consistent with Dr. Major's conclusion from his research in mice. So when I first received my project in the major lab, which is just to study a handful of lethal lines from the COMP project, that was about the time that I was transitioning from the clinic to do basic research. I remember there were two big wow moments. So the first one happened when I was studying a gene that is called ZinHit1, which is a protein coding gene. So almost no one had ever heard of it before because it is a very much understudied gene. And truly, we don't know a lot about it. But the surprise was the following. So when they're in culture, they look like this. So the top left corner is a mouse blastocyst, a typical mouse blastocyst. It's a control embryo. Consists of the ICM, surrounded by trophectoderm, and then further surrounded by the zona pellucida. And when we look at those ZinHit1 mutant embryos, meaning that in those embryos, there is no functioning ZinHit1 protein, all right? Those embryos look almost indistinguishable from the normal embryos. They can form a proper TE, proper ICM, and surrounded by zona pellucida. If you leave them in culture for a little longer, they had no problem growing bigger, and they will eventually become completely hatched or completely out of the zona. And most importantly, they are euploid embryos. Okay. However, the interesting part happens when we try to take a look at the implantation site directly in vivo. Remember, a perk with working with animal model is that we have the flexibility of dissecting and examining at almost any time point, right? So at a typical implantation site, the mouse embryo, like what Dr. Major just mentioned, will form this classic elongated egg cylinder shape right here. So which looks like a grain of rice, right? What we're looking at right now is a section of this embryo that is trying to implant into the surrounding maternal cells. And these cells are called the decidual cells. In the cartoon, those are the gray cells on the very outside. Remember, those are maternal cells. Those are the uterine wall. So with the use of immunofluorescence, which we can see different cell lineages in different colors, we're able to tell them apart. For example, OCT4 marks the epiblast, which will become the future fetus. CDX2 marks the extraembryonic ectoderm, which will contribute to the fetal portion of the placenta. And these two portions are surrounded by a single layer of GATA6 positive cells. These are called visceral endoderm, which you can see. Wait, let me move this bar real quick. In this cartoon, those are the purple cells that are right next to the blue cells. These are called the visceral endoderm, right? And then to the outside of these cells, we can see another population of GATA6 laminin double positive cells. And these cells are the bigger purple cells on the outside. These are called the parietal endoderm. These are the cells that are secreting laminin into the extracellular space, forming something called the Recker's membrane, right? Which is the maroon line you see in this cartoon. Now, one thing that stands out here is that normal embryos at this stage appear to be tightly anchored onto the uterus wall. And this is because of these yellow cells right between this embryo and the maternal cells. These yellow cells are called the trophoblast giant cells, and they are responsible for invading into the maternal tissue. So altogether, we don't see any space between this embryo and the maternal uterus. Now, with all these in mind, let's take a look at ZNH1 mutant embryos. So first of all, we notice that they are much smaller in size, finally, right? But if we look closely, they have proper cell lineage set up because they have OX4 positive portion, they have CDX2 positive portion, they have GATA6 and GATA6 laminin double positive cells. But very strikingly, those embryos is no longer anchored onto the uterine wall, but instead were left loosely hanging in this implantation crypt, indicated by the big space between this embryo and the maternal tissue. So our further research showed that ZNH1 mutant embryos were deficient in invading due to the fact that those yellow cells, let me remind you again, trophoblast giant cells, are functionally compromised. So what I just showed you is a vivid example of how a morphologically normal and euploid embryo failed to implant. And no matter how well we prepare the uterus, no matter how good of the media that you use in the culture, these embryos cannot implant. And this is due to the genetic feature of this embryo. Now, the second big wow moment to me I like to share today is when I was studying the sibling of ZNH1. It's actually called ZNH2. Again, ZNH2 mutant embryo means that in those embryos, ZNH2 protein is functionally null, right? So when in culture, we saw those mutant embryos have no problem forming blastocysts, right, of a comparable size to the control embryos. However, if you leave them longer in culture, instead of growing bigger and started to hatch, they all become collapsed and stuck in zona, and eventually they become dead. So then we use immunofluorescence to mark a core component of the spliceosome, which is called AFTA2. We notice that its nuclear localization is completely abolished in those mutant blastocysts. And remember, spliceosome is absolutely indispensable for nearly all mRNA splicing. So we expected to see a global change in the splicing efficiency. And indeed, from our RNA sequencing data, we observed a significant increase in the intronic regions. So this means that there are actually a lot of mRNA that is not being processed properly at this time. But remember, those mutant embryos can still form blastocysts. But we're not very surprised to see that those embryos won't survive long. So all these is telling us that ZNH2 mutant embryos, while exhibiting critical splicing failure, can still form blastocysts and still can be euploid, but they can quickly become not clinically usable. And of course, they will never be able to implant. And again, I wanted to remind us that this has nothing to do with what media you use, what incubator you use. Their deficiency is caused by their genetic features. So now this is getting exciting because these are not the only two genes that can cause embryonic lethality, right? Let me just remind you all about this COMP project that Jesse mentioned earlier. So far, this project has screened through over 8,000 genes, which is only about 1/3 of the entire mouse genome. But even with the current data, we know that about 1/4 of those genes, when one of them becomes functionally null, it will result in embryonic lethality. And we are talking about a little bit less than 2,000 genes here. And those lethality may present at the time of fertilization, at the time of implantation, gastrulation, organogenesis, et cetera, all sorts of miscarriages. Right. There's another group of about 8% of genes, which is over 600 genes. When one of them becomes functionally null, it will cause lethality in some embryos, but not in others, with a mechanism we don't quite understand at this point. So altogether, what we're looking at is actually a pool of over 2,500 genes that when one of them is acting up, we'd expect to see some level of infertility. And remember, COMP project is far from being done. So this list is expected to grow even longer. So that's all in mice. Given the high similarity shared between the mouse genome and the human genome, at this point, like Jesse mentioned, we're fairly confident that the human genome also contains this set of lethal genes. Those are the genes that, when functionally disturbed, make it impossible to achieve a live birth. Right? And the biggest question here is, what are those genes? And of course, more work is needed to be done on the human side specifically. But once we could identify them and understand the specific mutations that cause the failure, they could serve as powerful biomarkers. And this will not only help us with embryo selection in IVF, but also it will give us the much needed explanations for certain cases of infertility, where right now, what we can say is only sorry. We don't really understand why. Right. Okay, for the rest of the time, let's switch gears a little bit and let me introduce you to this very exciting translational project that we have developed in the lab, which actually started about 2 summers ago. It started in the summer, I remember. So the title is to investigate the potential use of DH1 in assisted hatching. And throughout this presentation, I'll be using DH1 as a placeholder in consideration of a pending patent disclosure. Thanks to everybody's effort here, from 2012 to 2021, the number of babies born in the United States using assisted reproductive technologies, ART, grew by 49%. And more than 99% of those ART procedures performed are IVF cases. In the US alone, more than 91,000 babies were born through IVF in the year 2022, making up 2.5% of all births that year. As of June 2023, global count of IVF babies has surpassed 12 million since the first IVF baby just born in 1978. So this is all telling us that the field of IVF has been rapidly progressing, showing no sign of slowing down. But however, if you are a patient today in the United States, you are expected to spend anywhere between $15,000 to $20,000 for a single IVF cycle. And remember, this number is a nationwide average. Okay. And the average number of cycles that are needed to become pregnant from IVF is estimated to be 2.5. So IVF is considered one of the most expensive medical procedures, especially when insurance coverage is still quite limited at this point. So moving forward, the future of IVF should become more efficient and more affordable. So now what we're looking at is a human blastocyst. I think many of us are very familiar with this. This embryo has 3 biggest components: the ICM, the TE, and the zona pellucida on the outside. This is how the normal development should look like. This blastocyst keep growing bigger and bigger, keeps expanding, and then eventually become out of zona. And this process is known as hatching. Right? However, not all embryos are like this. For example, this embryo on the right-hand side is clearly having some trouble hatching out. Right? And clinically, we call those embryos exhibiting hatching failure. As you can imagine, those embryos will have more difficulties in achieving a successful pregnancy. So indeed, hatching failure can significantly reduce the implantation rate, right? And clinically, hatching failure has been correlated with many, many factors that are frequently seen in current fertility centers, such as the advanced maternal age, the cause of infertility, and factors such as smoking, as well as external factors such as in vitro culture system. Well, unfortunately, this is an entire field. and another very commonly used procedure, which is called cryopreservation. Right. So in order to combat all these factors, clinicians and scientists have come up with different methods to assist hatching, which includes a chemical way where you use an acid to dissolve the zona from the outside. There's a mechanical way where you can mechanically drill a hole on the zona with the idea of letting the embryos come out more easily. And I think everybody's familiar with the laser-assisted hatching, which is shown here, is where you fire a few concentrated pulses of laser directly at the zona to create an opening for embryos to come out. So laser-assisted hatching is the most widely used assisted hatching method nowadays, which is estimated to be used in over 90% of fertility centers worldwide. Now, if you're a patient in the United States today, you are looking at an additional cost of $500 to $1,000 per cycle. However, laser-assisted hatching has mixed outcomes. So multiple recent randomized studies and meta-analyses has associated laser-assisted hatching with no significant increase or even lower birth rate. And this is the reason why, if you look up the latest guidelines from the three biggest ART authorities, which is ASRM based in the US, ASHRI based in the Europe, HFEA based in the UK, they have all stopped recommending laser-assisted hatching for all patients. So in order to improve IVF outcomes, we definitely need a more reliable and more efficient assisted hatching method. And this is where our research comes in. So in the next couple of slides, I'm going to show you some experiments that we've done in mice, and then I'll come back to human at the end. Although I cannot tell you what DH1 really is, but what I can tell you is that DH1 is naturally produced by mouse blastocysts as well as human blastocysts. In this following experiment, we wanted to use inhibitors with the intention to inhibit the function of the endogenous DH1 in mouse embryos. Okay. So we have our group A as control, group B as the inhibitor group. By the end of the culture, in the control group, we saw 4 embryos out of 8 started to hatch, while in group B, none of them were hatching. Right? So with this observation, it got us thinking, what if instead of using inhibitors, what if we just add more DH1 into the culture media? So now we have group A still as the control group, and group B is the group with exogenous DH1. And the result is quite surprising. Not only we saw that there's an increase in the hatching rate in all DH1 groups, but also their hatching rate is DH1 dose dependent, meaning that the more DH1 you have available in your media, the more hatching embryos you end up in having by the end of the culture. Okay. Remember about 10 minutes ago, I introduced ZINHIP2 mutant embryos to you. They are genetically modified embryos, right? And in the normal condition, none of those embryos could hatch on their own. So what we're seeing is DAPI signal. DAPI marks the nucleus, and then surrounded by this white dashed line, which indicates where the zona pellucida is. So none of those embryos can hatch on their own. However, in the presence of DH1, very surprisingly, we saw that some mutant embryos started to be able to hatch again. This is where this arrow is pointing to. This is saying that DH1 was able to rescue some of those hatching failure phenotypes that we see in mouse embryos. So now we wanted to ask, if DH1 assists hatching by promoting the overall proliferation. So in other words, do we see more cells in DH1 treated embryos by the end of the culture? Short answer is no, right? So this is the comparison of the total cell number across all doses, and we did not see any differences, even when we compare the 500 group to the control group. Right? However, when we look at the morphology of those embryos, we can clearly see that the embryos in the 500 group are a lot more advanced. So now we wonder, does DH1 change the lineage proportions? So in a mouse blastocyst, the same thing as in human blastocyst, the ICM has already begun to further differentiate into either epiblast or primitive endoderm, right? And with immunofluorescence, we can use different antibodies to look at those cells specifically. So we carefully compare their proportions and saw that there were no differences in the lineage setup. So the next question we went on asking is that, is DH1 treatment compatible with life? Because this is a translational project, we wanted to make sure that we could achieve live birth with it. So we set up embryo transfer experiments in mice. Embryos were retrieved at E2.5, which is a stage earlier than blastocyst, and then cultured either with or without DH1. And then by the time of blastocyst, they were transferred back into the female recipients. And then we wanted to see if DH1 treated embryos could lead to live pups. So the answer is yes. Well, we're still waiting on more numbers to come out, but now I can show you the pictures of the first 2 DH1 treated pups ever born in the world. This is when they look like when they were newborns, and this is what they look like when they were about 3 weeks old, and they have been running around and doing pretty fine. So to begin to optimize this protocol, we first evaluated whether a shorter exposure time would be sufficient, right? So we let the embryo exposed to DH1 for only 24 hours instead of the previous 48 hours. So we found that the exposure during the second half of the 24 hours, which initiated at around compact morula stage, could also significantly increase the hatching rate, as is shown in group B. And also, on the other hand, DH1 belongs to an evolutionarily conserved protein family, so we wanted to investigate whether other family members could similarly promote blastocyst hatching. So far, we have identified 2 other members that can significantly enhance the hatching rate. So at this point, we're calling them DH2 and DH3. And again, a lot more experiments are on the way. So all these are quite promising, but those were all done in mice. How about in humans? Does DH1 assisted hatching work with human embryos? So we are very lucky to be able to collaborate with NYU Langone Fertility Center to test this hypothesis in human embryos. Since they're very precious human samples, so the size is greatly limited for ethical reasons and availability, which makes me even more excited to share a case study we were able to conduct with these 5 frozen embryos produced from the same couple with a maternal age of 37 years old. So we carefully split the embryos into 2 groups, 2 embryos in the control media and 3 embryos in the media that's supplemented with DH1. So let's first take a look at the control embryos. So we can see that this first embryo is doing just fine, right? However, the second embryo seems to have some problem hatching out. It's definitely struggling. Now, let's take a look at the 3 embryos in the DH1 group. All of them were able to keep extending and keep pushing out and become entirely hatched by the end of the culture. So this is quite exciting. Altogether, if we pull all the embryos together, so we have set up 2 groups of experiment. So in the first group of experiments, we saw the embryos that were frozen at the cleavage stage, all right? And then expose those embryos to DH1 from compact morula stage and onwards to mimic the experiment we'd done in mice. And then we saw that the hatching rate was promoted significantly in DH1 group from 24% to 60%, right? In the second group of the experiments, because nowadays in the clinic, a lot of those embryos were frozen at the blastocyst stage instead. So in the second group, we used those frozen blastocysts and then exposed them to DH1 immediately after thawing. And we still get a solid 10% increase, still seeing the same trend. Although this difference is not statistically significant, which will require a lot more embryos, but nevertheless, we think that this is a very exciting start point. So overall, what I just presented to you is a potential assisted hatching reagent, which is going to be non-invasive, non-mechanical, and more affordable, which is going to be an easy addition to the current culture media that you use in the lab. We believe that DH1 facilitates cell-driven hatching because DH1, remember, is endogenously secreted by blastocysts. And as basic science researchers, we think that this project helped to understand the role of DH1 as well as its family members in the process of hatching. So with that, I'd like to thank my lab members, major lab members, Tremblay lab members, my committee members, my collaborators at NYU, and of course, funding agencies. And the last but not the least, I'm graduating soon and open to work. So if your clinic is hiring or if you know someone else is hiring, here's my contact info, my email and WeChat. And with all this, we'd like to take questions. Thank you very much.
Thank you so much, Doris.